Hi, everybody. Hi, if you're here, can you give me a little hi in the chat so I can see who we've got? Our year 13 lesson for today. Are we ready to start? We are, for some reason, it said stem cells on my title slide today, but it's all about mutations. Mutations and mutagenic agents and cancer a little bit as well. So if you can hear me, put a plus in the chat, give me a hey. Let me know who's here. I can't see anyone in the chat. What is going on? What is going on? Hey, good. I'm not just talking to myself. Hey, guys. Good, brilliant. I'm glad to have you here. Okay, let's get straight into it, shall we? Is anyone new? Is it anyone's first lesson? Let me know if it is. Hey, Abby. Hey, Priscilla J. Um, Hey, Miran. So we're going to do what is a mutation, first of all. Now, a lot of this is year 12, but we do it again in year 13 and we go into more detail. We link it to cancer and how mutations can lead to cancer. So first question for you, what would be your best definition of a mutation? Let's write that down first because it could come up as like a one marker. I mean, you should have known a decent-ish definition at GCSE. But yeah, think about maybe what you did in year 12. If you're doing AQA, you will have done this already in year 12. So what is a mutation? What are we going to say? And then the next question, if you're not confident with your definition, is can you give me any examples of mutagenic agents, which are basically things that increase the risk of mutation, they increase the frequency of mutations. Good. So Priscilla has given us a really good definition of a mutation. A random change in the DNA sequence. Change in the DNA sequence. I wouldn't say, let me just read yours. Emanuela, a change in the coding proteins. It's not directly a change in the protein, right? It's a change in the DNA itself. So let's say a random change in the, and we can be slightly more specific than just say DNA. We can say a random change in the base sequence of DNA. Now, Emanuela, that can obviously change the protein. It can change the amino acid sequence of a protein for sure, but the mutation itself is occurring in the DNA. Yeah, you've got a change in the base sequence of the DNA. Okay, does anyone know any examples of mutagenic agents then? Anything that if you expose yourself to it, or if it's present in your environment, it's going to increase the likelihood of a mutation occurring in the DNA. Good. So lots of you talking about radiation. So obviously radiation, exposure to radiation is a big one. There's different types of radiation that act as mutagenic agents. So UV radiation, X-rays, gamma rays. These are all mutagenic agents. So yeah, a carcinogen is by definition something that causes cancer. But yeah, obviously carcinogens, the way they do that, the way they kind of increase the risk of cancer is by causing mutations. So yeah, some of you, Felicity said smoking. So yeah, chemicals in cigarette smoke would be another example of a mutagenic agent. We could name a chemical. There's one called benzene. Don't know if you've heard of that one. That's the one I always remember because it's a short word. Some of these chemicals have mad names, and there's no way you're ever going to remember these mad names unless you're like a super chemist. But benzene is a nice short chemical that's found in cigarette smoke that is a mutagenic agent. So that's the one that I always remember. There's loads, yeah, there's loads of other ones. Another one that you might be aware of, some viruses are mutagens. So have we all heard of HPV? The human papilloma virus. So infection with that virus, it's classed as a mutagenic agent because it increases the risk of cervical cancer, which is caused by mutations in the DNA. So that virus is actually acting as a mutagen, a mutagenic agent, or a mutagen. Okay. I mean, I wouldn't worry too much about learning examples, but maybe try and have like 1 or 2 examples up your sleeve just in case they ask. Although I haven't seen them ask this much on A-level biology. They tend to focus on the different types of mutation and the effects of mutations, right? So should we get straight into that, and we'll see which ones you already know and which ones you need to go through a little bit more. So types of mutation. Have we all heard of these 3 types? Can you let me know in the chat? Because it might depend what exam board you're doing. You might not have done this yet, but we've got substitution, addition, and deletion. These are like the 3 most basic types of mutation. Okay, so most of you are saying yes, but there's a few people saying no. So let's go through and explain each one. The name of the type of mutation kind of tells you what that mutation type is, right? So a substitution mutation is where one base is substituted or switched for another. So one base is basically switched or substituted for another base. So if you look on my diagrams here, I've tried to show you an example of each of these mutations. So have a little look with me. If this was the original sequence, so the original sequence of bases in the DNA was TAACT. But here we've got a substitution because we've got TAACC. So can you see it's as simple as that nucleotide that had a thymine base has been substituted with a nucleotide with a cytosine base. So we've just switched one of the bases. So an error has obviously occurred during DNA replication. Instead of a T base, we've got a C base. So it's a random change in the DNA sequence. Yeah. Let's have a look at addition. Again, the name tells you what it is, but let's have a look at what's happened. If we compare our addition to the original base sequence, which is at the top, we've basically added in an extra nucleotide. So we've added in an extra base, an extra nucleotide with a cytosine base. And it might actually not just be one that's added in, there might be multiple bases added in. So I'm going to put the addition of 1 or more bases into the nucleotide sequence. Here we've just got the addition of 1 extra nucleotide. So we've ended up with 1 extra cytosine base in that sequence of bases, but you could see additions where multiple nucleotides are added in at multiple different points, right? And then deletion, again, the name gives it away. We've basically lost a nucleotide, we've lost a base, and if we compare it to the original sequence, we can see that it's a nucleotide with a thymine base that's been deleted. But it could be any, it could be multiple. So the loss of 1 or more bases from the nucleotide sequence. And all of these examples are changing the base sequence, right? Whether you're taking bases away, adding bases, switching bases, we are changing the base sequence of the DNA. So don't forget your basic definition of a mutation. I'm going to ask you a question that's not on here, but can anyone tell me the name of the enzyme that joins nucleotides together in DNA replication? What is the name of the enzyme that joins nucleotides together in DNA replication? Good, DNA polymerase. Okay, perfect. So basically, it's not worked as it should, right? The nucleotides have not been joined in the correct order, or a nucleotide's been joined when it shouldn't have been joined, or a nucleotide's been missed out. So yeah, something's gone wrong with DNA replication, okay? So these are the most three basic types of mutation. Let me just introduce a couple of other types and you can tell me whether you've heard of these before, whether you know what they are. So we've got duplication mutations and we've got inversion mutations. Now, who's heard of these ones before? Because most teachers probably wouldn't introduce these in year 12. They might mention them, I suppose, but it's just to kind of show you that there are other types. Okay, so more of you are saying no for these ones. You've not heard of these ones. Doesn't come up a lot on the A-level exam. It's just to show you that there are some other types of mutation. Again, the name kind of tells you. So here, I mean, this is also an example of addition. Yeah, it's also addition, but you've duplicated. So you've got AGGT in the original sequence, and then can you see in the mutated sequence, you've duplicated it. So you've got another AGGT. So you've duplicated, you've added in 4 bases. So it's also an example of addition, but you've basically duplicated the first part of that sequence. Have we missed a page? Have we? No, I don't think so. Unless you've got something else in your workbook that I've not got in my presentation, you'll have to let me know. Okay, so it's also addition, but it's addition of a sequence of bases. You've duplicated a sequence of bases. Inversion. Can anyone tell me what's happened here? Before I describe it to you, if you have a little look at the original sequence compared to the mutated sequence, what has gone on here? Can anyone describe it? It's quite hard to put into words. That's why I'm asking you to do it for me. 2 bases have swapped places. Let's have a look. I don't think 2, but yeah. That's a good way of saying it, A. A chunk of the sequence has been put in backwards. I think it's more than just 2 bases that have swapped. You've got a sequence of nucleotides or bases joined together in reverse order. Should we say it like that? I've never seen a question asking for a definition, by the way, so don't worry and think, how do we describe this? You're not going to be asked to describe it. You're just going to be asked to name it or spot it or highlight it. But yeah, in the original sequence, it went TGCA, and in the mutated sequence, it's gone ACGT. So 4 bases have been inserted in the reverse order. So you've not added any bases, you've not deleted any bases, you've still got the same number in total, but 4 of them were put in the reverse order. Okay, but you've still got the same total number of bases. Okay, but it still could affect the amino acid sequence. Remember, DNA codes for proteins. We need to learn this sentence off by heart. Let me write down one of the most important things to remember. The order of bases in DNA determines the order of amino acids in a protein. We all know that, yeah? We should do. So all of these examples, because we've changed the order of bases in some way, shape or form, we can potentially change the order of amino acids in a protein, okay? So important that we understand this and that we can use this phrasing in an exam question, whether it's about transcription or translation or mutations. We need to understand what DNA does, okay? Right, shall we do a little bit more writing then, guys? This is a year 12 question. Well, If you're AQA, I'd say this was a year 12 question, but I know with OCR, you tend to wait till year 13 to do mutations, so you might struggle a little bit more with this. But remembering the order of bases in DNA determines the order of amino acids in a protein. We need to remember that. So if there's a mutation in a gene in the DNA, it can lead to a protein being produced that has a different tertiary structure. It can lead to the production of a non-functional protein. Now, we're going to use an enzyme as an example because obviously enzymes are proteins. Enzymes are proteins, so they are coded for by DNA. Their tertiary structure, their whole shape, their active site, it's all determined by the order of bases in the DNA and the order of amino acids in that enzyme. So how can a mutation in the DNA lead to the production of a non-functional enzyme? Now, this could be like a 4 or 5 mark question on an A-level biology paper. So what kind of things are we going to include? We need to get all the way from the mutation, so the change in the DNA base sequence, to an enzyme that cannot function. What kind of things are we going to say? Let's try and get some bullet points going in the chat. So Zawad says it changes the shape of the active site. Good. It's no longer complementary to the specific substrate. Good. What is a tertiary structure? So the tertiary structure is like the complex 3D structure of the protein. Now, enzymes have what we call globular tertiary structures. Have you heard that before? They tend to be more globular, more rounded, yeah? Some proteins are more fibrous, things like collagen or keratin. They tend to be like long and thin. They have a long, thin tertiary structure. But enzymes and things like hormones have a globular, like a more spherical tertiary structure. But it's basically the complex folding of that protein to give it its overall 3D shape. Yeah, hemoglobin is globular as well. Hemoglobin is made from 4 polypeptide chains. They all come together to give it an overall globular tertiary structure and quaternary structure. So Grace says you've got a different sequence of bases leads to different amino acids forming the polypeptide. This leads to different bonds in the tertiary structure. Very well done. Can we name any of those bonds, Grace? Can we name any bonds that hold the tertiary structure together? Felicity says the mutation can change the order of bases in DNA, which can change the amino acid sequence in a protein. This can affect the tertiary structure of the protein, and the active site will no longer be complementary. Yeah, insulin has a globular structure. The sequence of amino acid may change, which would change the R groups that are present. Nice. Jay has even named, and Priscilla and Grace, yeah, you've named some of the bonds. Just Grace, the disulfide bonds are the covalent bonds, okay? Disulfide bonds are covalent bonds. That's the same thing that you're talking about there. Okay, right, let's get this down. I'm going to use a lot of your answers because they are good. If we kind of put them together, we can get all of the marks. So let's start with, there is, and I'm going to do this really simply because this is a key idea in biology. So there is a change in the base sequence of DNA. So we're telling the examiner what a mutation is, okay? Because they've not told us in the question, they've just said there's a mutation. So we're saying, well, that would change the base sequence of the DNA, okay? And this can change the amino acid sequence of the enzyme because the enzyme is a protein. Guys, what is the amino acid sequence called? What term do we give it in protein structure? the basic order of amino acids is called the what structure? Thanks, that the primary structure. So you could say, instead of saying this can change the amino acid sequence, you could say this will change the primary structure. Yeah, same thing, same marking point, because primary structure is just the order of amino acids. Okay, someone said as well that this means you'll have different R groups. We can put that in. It might be worth a mark, it might not, but this will change the order of the R groups. Now remember, all amino acids have a different R group. Yeah, it's the variable side chain. So if you've got a different order of amino acids, you've got different R groups. You've got a different order of R groups. Okay, we have to talk about bonds, okay? Because we're going to start to think about the tertiary structure. Whenever we talk about the tertiary structure, we need to name at least one bond that holds the tertiary structure together. What are R groups? Do you remember the structure of an amino acid? Here's the general structure of an amino acid. So it's this group here. So all amino acids have the same parts here. So they all have the amine group, the central carbon, the hydrogen and the carboxyl group, but they all have a different R group. That's what makes each amino acid different. Yeah, there are 20 different amino acids, all got different R groups. So if we're changing the order of amino acids, we're basically changing the order of those R groups, okay, the variable side chains. Okay, so if we change the order of the R groups, this will change the position of bonds holding the tertiary structure together. And I'm going to say, for example, because quite often they will give you a mark for naming a bond. So for example, ionic bonds will form in different places, hydrogen bonds will form in different places, and disulfide bonds will form in different places. Okay, so you don't have to name all three, but name at least one bond type that you know you find in the tertiary structure. And disulfide bonds are covalent bonds. Okay, so you could say covalent bonds. So if we're changing the bonds that hold the tertiary structure together, then the tertiary structure changes shape. We've got a different tertiary structure. Yeah, you could say hydrophobic and hydrophilic interactions as well. They're obviously a lot weaker. They're just interactions, they're not bonds, but you could mention those as well. Although I would always mention one of the main ones as well, ZG. I'd always mention ionic, hydrogen or disulfide as well. So change the tertiary structure, and then because this is an enzyme, we can go all the way to saying it changes the shape of the active site and all the way to saying substrate is no longer complementary. To active site. Or you could say substrate can no longer bind to active site, or enzyme substrate complexes can no longer form. If we say covalent bonds instead of disulfide bonds, will we still get a mark? That's a really good question. I personally wouldn't, because covalent bonds, that's less specific, right? Because there are all different types of covalent bonds. Like peptide bonds between amino acids are also covalent bonds. So it's better if you say disulfide bonds, I'm going to be honest. Why haven't we talked about the secondary structure? Right, that's a really good question. Because the secondary structure is not determined by the order of amino acids. okay? The secondary structure, which is your beta-pleated sheets and your alpha helices, that is only held together by interactions between these parts of the amino acid, the amine group and the carboxyl group. So it doesn't matter what the order of amino acids is, because all amino acids have the same amine group and the same carboxyl group. So the secondary structure is not determined by the order of amino acids, okay? But the tertiary structure is, because the tertiary structure is held together by bonds between the R groups. Does that make sense, Sienna? The tertiary structure is determined by the order of amino acids. So if you change the order of amino acids, you'll change the tertiary structure. But the secondary structure, it doesn't matter what the order of amino acids is. Let me know if that makes sense. I mean, they're not going to penalise you if you talk about the secondary structure, but it's not relevant, okay? Because changing the order of amino acids doesn't affect the secondary structure. It affects the tertiary structure. Okay? Just try to remember that the tertiary structure, I mean, I'll put a little note for you, Sienna. Tertiary structure is determined by the order of amino acids. It's determined by the order of the R groups, okay? So that's the one we need to focus on. And to be honest, in biology, we always focus on the tertiary structure. It is like the most important level of protein structure. Okay, personally, I love this question. Like if I got a question like this on an A-level exam, I would just think, perfect. I know I can get all the marks. Even if this was 5 or 6 marks, I can get them. I love this. I love this whole story going from DNA to amino acid sequence to tertiary structure, and even better if it's an enzyme, because then I can also link it to the active site and the enzyme substrate complexes. It's such a nice story. Don't worry, Sienna. Okay. Right. Hopefully that kind of was relatively easy. It's just remembering it, right? It's just revision from year 12 and all of your protein structure stuff. Let's move on. I just thought I'd throw something else in there, something that maybe you've not heard about yet. Does anyone know what a missense and a nonsense mutation is? Has anyone heard of these terms before? Missense and nonsense? No? No? Okay. You've heard of them, but you don't know what they mean. Okay. So I'll just give you some definitions. Nonsense, like it has no effect. Not quite. A silent mutation is one that has no effect. A silent mutation. Missense is when, let me put this down as concisely as I can. The mutation causes a different amino acid to be coded for. Okay, so maybe it was meant to be maybe the base sequence was meant to be AGT, but now it's TGT and it codes for a different amino acid. So it was meant to be one amino acid in the sequence, but you've ended up with a different amino acid in the sequence. Yes, Johan. A nonsense mutation is where the mutation now, instead of coding for amino acid, the mutation now does not code for an amino acid, but is a stop codon. Did you guys know that there are 3 stop codons? 3 stop codons, 3 sets of bases that mark the end of the polypeptide. Can't remember what they are off the top of my head, but there's basically 3 triplets or 3 codons that don't code for an amino acid at all. They actually mark the end of a polypeptide. They tell the ribosome to stop translation. They mark the end of the polypeptide. Now, you can get a mutation where, I don't know, it was meant to be EG, why do I keep saying E? It was meant to be AGT, but now it's a stop codon. So not only are you going to put in the wrong amino acid, but you're going to have no amino acid put in at all, and the protein is going to stop there. Yeah? What are the stop codons, guys? Is anyone? Oh, Annie. I was just about to ask, can someone find out? Because I can't remember them off the top of my head. So UAA is a stop codon. UAG is a stop codon. And UGA is a stop codon. Now, they're obviously in RNA. I know they're in RNA, the ones you've given me, because you've got U's in there. So in DNA, the stop triplets would be ATT, ATC, and ACT. So basically, if you get any of these in DNA, then you are marking the end of the polypeptide. You're saying there's no amino acid to go here. This is the end of the polypeptide. You don't have to know them, by the way. Methionine, Zada, methionine is a start codon. That's coded for by a start codon. That's always the first amino acid that goes in, methionine. Just one sienna. So there just needs to be one of these triplets in DNA or one of these codons in RNA. I'm going to rub them off because I don't want to make you think you have to learn them. That was just for me to see if I could remember. Yeah, there only needs to be one. So there's one of these triplets in the DNA that marks the end of the gene, basically, the end of the protein that we're going to code for. And then that stop codon gets put into the mRNA, and that marks the end of the mRNA and the end of the polypeptide. But imagine you get a mutation, and the mutation now is a stop codon. That protein is never going to get finished. You're going to end up with a really short polypeptide that is finished way too soon and has way too few amino acids. Could a uracil base ever accidentally join to a DNA strand? I've never thought about that before. Not that I've heard of, but now I'm thinking about it, I'm thinking, why couldn't that happen? Maybe it's to do with the sugar. You know, because RNA nucleotides have ribose sugar. So I'm guessing that DNA polymerase would not be able to join that in because they'd have to join like a ribose sugar next to a deoxyribose sugar. So I don't think it would work. So what would happen to the mutated polypeptide? Let's have a little think, Jay. Imagine, I don't know, the protein was meant to be this long, but there was a mutation in, let's say, the 4th codon, and it coded for a stop codon. So the protein is only going to be this long. So it's going to have a huge effect, right? It's going to get stopped early. So it's not going to have any of these amino acids. So the tertiary structure is not going to form in the way it should. I'm pretty certain this protein is not going to be able to function because the polypeptide would be shorter. The tertiary structure would be different. I suppose it depends where the mutation is though. Think about it like this. If that mutation is right near the end anyway, it might not have as big an impact. Like if the mutation was further along, then the polypeptide might only be 1 or 2 amino acids short. Might not have a big impact. But if the mutation is very early on, and the polypeptide ends up way shorter than it was meant to be, that protein is not going to be able to function. whatever it is. Yeah. The tertiary structure would not form correctly, it wouldn't be able to function. But it does depend where the mutation is, because if it is pretty much towards the end anyway, you just get the stop codon like 1 or 2 codons earlier, might not have a big effect. Okay. Quite interesting to think about, really. Right. Addition, deletion, and duplication, which is also an example of addition. They nearly always change the amino acid sequence because they cause a frame shift. Who can give me a definition of a frame shift? I'm going to try and give you an example. Who knows what a frame shift is? Yeah, Grace, that's not a bad definition, actually. I'm trying to show you it here. Grace says all the bases after the mutation move. Yeah, what I've tried to do here is I've added in a base. I've added in thymine. And can you see, because I've added a base in, all the subsequent triplets, or you can call them codons, it's up to you, all the subsequent triplets are now different because we've added a T in. So the second triplet now reads TCC, but it has a knock-on effect, right? Because all the bases have been shifted by 1. So now the next triplet is also affected, and the next triplet is also affected. It's like you've shifted the reading frame. That's why it's called a frame shift. All the subsequent triplets are impacted. Yeah, when the sequence of bases moves according to the number of bases added or removed. Yeah, that's not a bad definition either. Let me write this down. All of the subsequent triplets or codons are altered or affected. So it can potentially change all subsequent amino acids. When I say subsequent, I mean after the mutation. Okay. Is proteome all of the proteins in an organism? Yeah, a better definition though would be to say the range of proteins made by a cell, but they do allow the range of proteins made by an organism. But you need to convey, don't just say all the proteins, say the range of proteins or all the different proteins. All the different proteins made by an organism. Why do bases come in triplets? It's just how the genetic code works. It's called a triplet code. So 3 bases code for 1 amino acid. So 3 bases code for 1 amino acid. So 3 bases in DNA is called a triplet. And then 3 bases in mRNA is called a codon. I don't know if you realised the difference, but for A-level, we don't really mind. You can call them triplets, you can call them codons, but strictly speaking, when we're looking at DNA, 3 bases is called a triplet. And then when we make the mRNA during transcription, the 3 bases gets called a codon. But you can usually just use either, right? But strictly speaking, it's a triplet on DNA and a codon on mRNA. But hopefully my diagram has helped you there, because you can see not only has the mutation changed the second triplet, but it's had a knock-on effect to the subsequent triplets. So the third triplet has changed and the fourth triplet has changed. So potentially you can alter all of the amino acids from that point. okay? Because all of the triplets are going to be altered. If you just add in a base, or you take away a base, or you add in 2 bases, or you take away 2 bases, you're going to affect all the subsequent amino acids, okay? Okay. Now, we mentioned this briefly before, but there is such thing as a silent mutation, okay? So we've had missense, where it codes for a different amino acid. We've had nonsense, where it codes for a stop triplet or a stop codon. Now we've got silent mutations. As the name suggests, a silent mutation does not affect the protein. It has no effect, okay? So not all mutations result in a change in the amino acid sequence. And silent mutations have no effect on the protein. Basically, even though there's been a mutation, the protein can still function. It still works. It's still fine. So the main reason for this is the genetic code is degenerate. So you can have a mutation and it will not affect the protein. So what does degenerate mean, guys? We always say in year 12, the genetic code is degenerate. Good, Sienna. Good, Felicity. Good. Multiple triplets code. I like that one. Multiple, or you could just say more than one triplet or codon can code for the same amino acid. So there might be a mutation. It might be a substitution mutation. Let's find an example here. You can find loads in this table. You can have a little look after the lesson and see what others there are. Look, maybe the original DNA had this triplet and there's a substitution mutation and now we've got this triplet. No bother, no problem, because they both code for an amino acid called Phe, which I think is phenylalanine. Doesn't matter. There's some, like, look here, look at arginine. CGT, CGC, CGA, CGG. Any of those, if any of those were substituted, it would still code for arginine. So because the genetic code is degenerate, you can have silent mutations. Yes, you change the base sequence, but it still codes for the same amino acid. Swaro's just asked, what's the difference between degenerate and universal? So degenerate is where you can have more than one triplet codes for the same amino acid. Can anyone tell us what universal means? Felicity says the same triplets or codons code for the same amino acids regardless of the organism. That is a perfect definition. Swaro, write down what Felicity said. Universal means the same triplets or the same codons code for the same amino acid in all living things. So if we are looking at, let's take an example. CCT codes for leucine. In humans, in wasps, in bacteria, in grass plants, in every single living thing, if there is that sequence of bases, it will code for the same amino acid. It's evidence for evolution. Yeah, all living things, same triplets code for the same amino acids. It's like really good evidence for evolution, isn't it? Other reasons why mutations may not affect the function of a protein. Think about where the mutation may have occurred. See if you've done this before. Again, if you're AQA, you mention this in year 12. But if you're OCR, you don't mention this till year 13, just a slightly different order of doing things, but you will all do it. So think about where the mutation might occur and think, look, if there's a mutation in this bit of the DNA, it will have no effect on the protein. Good, Grace. Good, Felicity. You guys are on fire. Have you been doing some sneaky revision? Sneaky revision over the summer? You've got good memories. So what about if the mutation was in an intron? It's going to have no effect on the protein because introns get removed. Yeah? They get spliced out anyway. So you're going to cut out the region that had the mutation in. So it's no bother. Doesn't matter. So they're the two main things to remember with a silent mutation, okay? It might be that the mutation is in an intron. You could give that as a reason for why the mutation has no effect on the protein. Or it might be because the genetic code is degenerate. Because even though there's a mutation, it still codes for the same amino acid. Okay, is this making sense, guys? Because I know this lesson is kind of getting gradually harder. Is this making sense? Because we've got all different types of mutations going on in our heads now, haven't we? We've got silent mutations that have no effect. We've got mutations that change the amino acid for a different amino acid, change the amino acid to a stop codon. Some amino acids cause a frame shift. So we need to try and like have all of these different types. Okay. And don't worry, because you're obviously going to do this again in year 13. Even if you're AQA, you do go over this again in year 13. Is intron non-coding? Yes. So obviously in DNA, some parts of the DNA are what we call non-coding parts. They don't actually code for amino acids in the final protein. And those parts just get removed anyway. So after transcription, they get removed from the mRNA. So it doesn't matter really if there's a mutation in there. Okay, let's have a go at a few questions then, guys. Oh, this looks like a long one. We've got sickle cell disease is a group of inherited disorders. So genetic disorders is another way to think of those. People with sickle cell disease, have we heard of it? Sickle cell disease or sickle cell anemia? They have sickle-shaped red blood cells. So instead of their red blood cells being like this, their red blood cells are kind of like this. Yeah? A single base substitution can cause this. This is true, by the way. We've not just made this up. So sickle cell anemia is caused by just one base being substituted in the gene that codes for hemoglobin. Okay. This mutation causes a change in the structure of one of the polypeptide chains in hemoglobin. Hemoglobin is actually made up of like 4 polypeptide chains. I'll just try and draw it here. 4 polypeptide chains. So if there's a mutation in one of the genes, just one single base is substituted, it changes the shape of one of these polypeptides. And then the hemoglobin is the wrong shape. And then the red blood cell ends up being the wrong shape. Explain how a single base substitution can cause a change in the structure of a polypeptide. Okay. What is W for win? So explain how a single base substitution. So this is what we need to do. Okay. We don't need to overthink it. Doesn't even really matter what the example is. We just need to say the same ideas. So if we've got a single base substitution, we have changed the order of bases in DNA. Even if we've just switched one, we've still changed the order of bases. So we've then going to change the primary structure of the protein. Or you could say a different amino acid in the primary structure. Okay, so you're basically going to have a different amino acid in that sequence of amino acids. So you've changed the sequence of amino acids. You could say you've changed the primary structure, or you've now got a different amino acid in the primary structure. And where are we going to go to next? Because this is 3 marks. What other level of protein structure do we always talk about in biology? If we change the primary structure, we've got a different amino acid in there, in that sequence. If we change the primary structure, what other level of protein structure are we going to alter? Good. Changes the tertiary structure. And again, I personally would always say as bonds, such as ionic, hydrogen, or disulfide, form in different places. I always mention the bonds for the tertiary structure because quite often you need both things to get that 3rd mark. So even though it's a single base substitution, obviously we know it's not silent. It must have changed the amino acid because it's resulted in a really serious disease. It's resulted in non-functional hemoglobin. So we know it's not silent. We know that substitution must have changed the amino acid. Okay, so we've changed the order of bases. It must have changed the amino acid. So we've got a different sequence of amino acid, which must have changed the tertiary structure. We must have these ionic or hydrogen or disulfide bonds forming in different places, different tertiary structure. That's why hemoglobin is not the right shape and it's non-functional. It basically cannot carry as much oxygen as it should. Okay, that's the end result of sickle cell anemia. The red blood cells cannot carry enough oxygen. Okay. Right, suggest how a mutation can lead to the production of a protein that has 1 amino acid missing. Oh, this is a good question. Let's see who's using their brains. You've got to think outside the box. You've got to use a bit of common sense. So we know that the protein just has 1 whole amino acid missing. What must have happened in the DNA to end up with 1 amino acid missing from the protein? Okay. Emanuela's got 1 mark. We are missing an amino acid, so we've clearly lost some bases. We've deleted some bases. Ooh, stop codon. Suppose it could have been. It would have, for only 1 amino acid to be missing though, you would have had the stop codon just 1 codon too early. We're not getting the 2nd mark though, are we? To lose an amino acid, you've obviously lost some bases. You've deleted some bases. Grace is on the right lines. Oh, that's it. Zada's got it. It must have been 3 bases deleted as there's no frame shift occurred. Do we get that? I feel like not many people are getting that in the chat. If you're only missing 1 amino acid, remember 3 bases code for 1 amino acid. So what if we delete 3 bases? If we delete 3 bases, we're going to lose a whole amino acid. But it won't affect any of the other amino acids, will it? Because it won't cause a frame shift. If you delete 3 bases, let's do an example. Yeah, if we deleted these 3 bases, we would lose one amino acid from the final protein, but the rest of it would be fine because you've not caused a frame shift. CAC is still read in the same way. So I think that's how you could end up with a mutation where just one amino acid is lost from the protein. You'd have to delete bases, but you'd have to delete 3 bases in order to remove one amino acid. Does that make sense? Can't be in an intron, this one, because you have lost an amino acid. So it must have not been in an intron. It must be in an exon. Because it has affected the protein, right? You have lost an amino acid. So you've definitely got a deletion here. But it must be a deletion of 3 bases. I think what we might do, so I think I've overplanned again, haven't I? I'm going to let you guys read this. There's nothing really that I need to teach here. This is some kind of extra year 13 stuff. But what you can do, if you've got time today, have a little read. This is where I'm talking about how a mutation can cause cancer, okay? But I've given you all the notes. So there's a gene that's called a tumor suppressor gene. And if there's a mutation in a tumor suppressor gene, it can cause cancer, uncontrollable cell division. But this diagram basically explains all of that to you, okay? So because we're running out of time and I'm not sure what other lessons you've got today, I'm going to let you read through that to try and learn a little bit more about how it can lead to uncontrollable cell division, a tumor, cancer, if there's a mutation in this particular gene. But what I'm going to do with you to finish, I think, is a couple of maths questions. Where's my calculator? Guys, if you've got a calculator or your phone calculator, do you want to grab it now? I'm going to grab mine. I just want to see whether you can do these kind of questions, okay? So they're maths questions, but they are linked to this topic. So we've got a tumour cell that divides every 8 hours. Starting with one of these cells, how many tumour cells would be present after 4 weeks? Do we know how to do these questions? Do we know how to do these questions? Shall I give you a bit of time to get a calculator? To the power of one another. You are right, we are going to use a power. I'm just working out to begin with, in 4 weeks, how many divisions would take place. So 4 weeks is obviously 28 days. If we multiply 28 days by 24 hours, 4 weeks is 672 hours. So how many divisions can take place? If it takes 8 hours for one division, how many divisions can take place in 672 hours? Good. A few of you have got that right. So we've got 84 divisions. Yeah. So what we do is 2 to the power of 84. And I'll explain that a little bit more. We just work it out. 2 to the power of 84. And it says give your answer in standard form because it's going to be such a massive number. I mean, you could do 1 times 2 to the power of 24, but obviously you don't need to. So the reason I'm doing this is we're starting with one cell. So good, Amberjeet. I'm starting with one cell, so I could do 1 times, but obviously I'm just timesing it by 1, so I don't have to. But I'm doing times 2 to the power of 84 because I'm doubling each time. So each time one cell divides into 2 cells, oops, I'm doubling the number of cells, yeah? So we're doing 2 and we're doubling it 84 times because 84 divisions can take place in 672 hours. So it's 1 times 2 to the power of 84 or just 2 to the power of 84. So I'm doubling it 84 times, if that makes sense. I'm going to 2 to 4 to 8 to 16 to 32 to 64, 128. I keep doubling 84 times. So it's 1 times 2 to the power of 84. Okay with that? A few of you have got the right answer. Should we have a go at this one? Guys, have any of you got another lesson at 1 o'clock? So I don't want to run over if you have. Has anyone got another lesson? No? Okay. Okay, let's finish with this then. I don't mind running over a little bit if you don't. So in the UK in 2016, there were 525,048 deaths. That's the total number of deaths. Cancer caused 30.4% of all deaths, and throat cancer caused 5% of all deaths from cancer. So calculate the mean number of people who died of throat cancer per month in 2016. So the first thing I'm going to do, I've got the total number of deaths. I'm going to find 30.4% of that to find how many deaths were caused by cancer. So I'm going to do 525,048 times 0.304. Is that how you guys work out percentages, or do you do it differently? 59, I'm just looking. 1596. Oh, how are you guys so fast? Now I'm gonna continue to step it out for those people that don't get it. Now I'm gonna find 5% of 1596.1.46 because I know 15,961.46 deaths were from cancer, and I know that 5% of those deaths were from throat cancer. So now I'm going to find 5% of that. Let's hope I'm doing this right, guys, and I get the same answer as you. So now I know how many people actually died from throat cancer, but that was in a year, right? And I want it per month. So I'm going to divide that by 12. Am I doing this right? Yeah, well, I'm getting the same as you. 665. How did you do that so quickly? So 665 people per month died from throat cancer. Quite an easy maths question. It's just actually reading the question, isn't it? Because you've got the total number of deaths. You need to work out how many from cancer, then how many of those were from throat cancer, and then divide it by 12 so you've got it per month. Guys, do you do A-level maths? Who does A-level maths here? Yeah, that's why you're faster than me. ZG doesn't, but everyone else is saying, yep. A doesn't, Johan doesn't. Well, I hope that was okay. That was just a percentage one, right? So nothing too bad. But it's always good to do some maths with biology because there's always going to be 10% of your marks are going to be maths, right? So you've got to prepare for it. I always think just like whatever maths questions you can find online, just always have a go. Even if you don't like maths, it's good to do as many as you can. Okay, right, guys, only a little bit over. So hopefully you were okay with that. Have a go at your homework. You've told me that you've got no other lesson right now, so you can do your homework immediately. And also have a little read back through the lesson notes on cancer, because it is interesting to find out how mutations can directly cause cancer. Have a little read through, and then I'm going to see you again tomorrow for lesson 4. I hope it's been useful today, guys. Thank you. Have a good one. See you tomorrow.